Good day, everyone. Today, I'll be presenting on cell injury and death, based on Chapter 2 of the BRS Pathology book. This chapter focuses on the causes of cellular injury, the mechanisms involved, and the different types of cell death, including necrosis and apoptosis. Objectives. By the end of this presentation, we'll have a clearer understanding of how cells respond to injury and how the processes of necrosis and apoptosis differ. Cell injury. The first concept we'll explore is cell injury. This occurs when a cell can no longer maintain homeostasis due to either external or internal stress, leading to damage. Reversible versus irreversible injury. Reversible injury happens when the cell can recover if the stress is removed in time. Irreversible injury is more severe and leads to cell death, which occurs through necrosis or apoptosis. This is a key distinction we will explore later. Causes for cell injury: physical agents. These include trauma, temperature extremes, and radiation. These external factors can cause direct mechanical injury or thermal damage. Chemical agents. Chemical exposure, such as drugs, toxins, and pollutants, can harm cells. For example, alcohol can damage liver cells, while carbon monoxide can disrupt oxygen transport. Biological agents. Infections by bacteria, viruses, fungi, and parasites can cause direct injury to cells. Immunological reactions. Immune responses, like autoimmune diseases or hypersensitivity reactions, can damage healthy cells. Nutritional imbalances. Both deficiencies, like vitamin deficiency, and excess nutrients, like obesity, can impair cellular function. Cellular response to injury. Adaptive responses. When a cell faces stress, it might adapt in various ways to survive. Hypertrophy. The cell increases in size to meet higher demands. Hyperplasia. There's an increase in cell number in response to stress. Atrophy. The cell shrinks due to decreased function or supply of nutrients. Metaplasia. One cell type replaces another as a form of adaptation. Injury responses. While these adaptations can help a cell survive, if the injury is too severe or prolonged, it can lead to irreversible damage. Mechanisms of cellular injury: hypoxia and ischemia. Both of these conditions deprive the cell of oxygen and nutrients, leading to mitochondrial dysfunction and loss of ATP. This is a major cause of cellular injury, particularly in organs like the heart and brain. Chemical injury. Chemicals can directly damage membranes or indirectly through the formation of reactive free radicals, which cause widespread damage to proteins, lipids, and DNA. Infectious agents. Pathogens such as bacteria, viruses, and parasites can cause direct cellular injury through invasion and the release of toxins. Immunologic injury. The immune system, while designed to protect the body, can sometimes attack normal, healthy tissues, resulting in inflammation and further damage. Reversible cell injury. Characteristics of reversible injury. Cellular swelling. Cells may swell due to a failure in ion pumps, which causes an influx of water. Fatty change. Accumulation of lipid droplets in the cytoplasm occurs, particularly in the liver and heart. Mitochondrial swelling. The mitochondria may swell due to ATP loss and ion pump failure. Reversibility. If the stress causing the injury is removed in time, the cell can return to normal. This is the hallmark of reversible injury, as long as the damage hasn't progressed too far. Irreversible cell injury. Irreversible injury. If the damage is too severe or persistent, the injury becomes irreversible, leading to cell death. Types of cell death. Necrosis. Uncontrolled cell death due to acute injury, which results in inflammation. Apoptosis. Programmed cell death, which is a controlled process, typically not associated with inflammation. Key features of necrosis. Loss of plasma membrane integrity. Leads to leakage of cellular contents and inflammation. Cell swelling. Due to loss of osmotic control, resulting in damage to organelles. Cytoplasmic changes. Eosinophilic staining due to protein denaturation. Nuclear changes. Pycnosis, condensation, karyorrhexis, fragmentation, and karyolysis. Dissolution. Enzyme leakage. Digestive enzymes are released, further damaging tissue. Inflammation. Triggered by spilled cellular contents, causing surrounding tissue damage. No apoptotic features. Unlike apoptosis, necrosis is uncontrolled and causes rupture of cells. These features collectively define necrosis as a pathological process that causes widespread tissue damage and inflammation. Necrosis types of necrosis. Coagulative necrosis. Seen in ischemic injuries, like myocardial infarction, where tissue retains its structure but cell death occurs. Liquefactive necrosis. Occurs in tissues like the brain and in abscesses, where cells are digested and transformed into a liquid state. Caseous necrosis. Common in tuberculosis, where a cheese-like material forms due to granulomatous inflammation. Fat necrosis. Occurs in adipose tissue, often due to trauma or pancreatitis, where fat cells break down. Fibrinoid necrosis. Seen in blood vessels, especially in autoimmune conditions like vasculitis. Key features of apoptosis: cell shrinkage. The cell reduces in size. Chromatin condensation. Nuclear chromatin becomes tightly packed. Membrane blebbing. The cell membrane forms small protrusions, blebs. DNA fragmentation. DNA breaks into smaller fragments in a controlled manner. Formation of apoptotic bodies. The cell breaks into membrane-bound fragments. No inflammation. Cellular contents are contained, preventing inflammation. Energy-dependent process. Apoptosis requires ATP for execution. Apoptosis is a programmed, controlled cell death process that eliminates unwanted or damaged cells without causing inflammation. Apoptosis. Definition of apoptosis. Apoptosis is a programmed form of cell death that happens in response to specific signals. It occurs without inflammation and helps in the removal of damaged or unwanted cells. Key features. The process involves DNA fragmentation, cell shrinkage, and membrane blebbing, all regulated by caspases, the enzymes responsible for dismantling the cell. Physiological apoptosis. This is part of normal processes like embryogenesis, immune system development, and tissue turnover. Pathological apoptosis. Abnormal apoptosis can occur in diseases like cancer, neurodegenerative diseases, and viral infections. Mechanisms of apoptosis. Intrinsic pathway. This pathway is activated by cellular stress such as DNA damage or hypoxia. Mitochondria release pro-apoptotic factors, which initiate apoptosis. Extrinsic pathway, triggered by external signals, typically from death ligands, e.g., Fos ligand, binding to receptors like Fos on the cell surface. Apoptosis is executed via two central apoptotic pathways, the extrinsic and the intrinsic pathways. Both apoptotic pathways converge on the same terminal or execution pathway. The extrinsic pathway begins with the attachment of extracellular ligands to the extracellular domain of transmembrane receptors, whereas mitochondrial apoptosis is initiated by several intracellular stimuli. Initiator caspase of both pathways, caspase 8 and 9, 10, can catalyze the proteolytic maturation of effector caspases, such as caspase 3, 6, 7, which lead to initiation of a caspase cascade and finally result in demolition of the cell. DNA damage triggers apoptosis by activation of the tumor suppressor protein p53. Apoptosis versus necrosis. Speaker notes. Necrosis. This process is uncontrolled and usually caused by ischemia, toxins, or trauma. It leads to inflammation, and the cell ruptures, releasing its contents into the surrounding tissue. Apoptosis. Apoptosis is a regulated, controlled process. It doesn't cause inflammation, as the cell fragments are cleared by phagocytes without damaging surrounding tissues. On comparison, this slide will help you clearly differentiate between necrosis and apoptosis in terms of their mechanisms, outcomes, and effects on surrounding tissues. Consequences of cell injury. Cellular adaptations. Cells may either recover or undergo permanent changes depending on the injury type and severity. Tissue repair. Repair can involve either the regeneration of normal tissue or the formation of scar tissue, fibrosis, depending on the extent of the damage. Pathological conditions. Prolonged cell injury can lead to chronic inflammation, fibrosis, or even cancerous transformations in the tissue. Summary of key points to wrap up. Reversible injury can lead to recovery if the stressor is removed. Irreversible injury leads to cell death, which can occur via necrosis or apoptosis. Necrosis involves uncontrolled cell death with inflammation, while apoptosis is a controlled, non-inflammatory process. The consequences of cell injury can manifest as acute events like necrosis or chronic conditions like fibrosis and cancer. This presentation was based primarily on BRS Pathology Chapter 2, but additional pathology resources may have been consulted for detailed insights. Please refer to the books for more information if needed.